And they told you guys there'll be videos, video lectures and things like that. All right? You guys can sit and watch them again or, use them as sleep aids at times. All right? I just wanted to go through a few things. Have you guys all been able to access Blackboard and everything, okay, with the syllabus, block materials for Exam 1? All right? Okay. All right, well, just get into it. Try to get out of here a little early so you guys can go relax. So this Biomedical Foundations, the course is mainly a hodgepodge of things some of the things that you probably learned as an undergrad, basic biology, some basic chemistry, okay? There's some microbiology. All you guys take microbiology? Yes? Okay. So microbiology, there's some cell biology, and then there's some biochemistry thrown in. There's some metabolism at the end that students don't seem to like. I try to make it as fun as I can, but it's hard. All right. The instructors for this class, you'll see me most of the time. There's our one pharmacy practice person comes in and talks about lab tests is Dr. Pound. Okay. Dr. Toth, he'll do a lot of the physiology, little bit of physiology. Dr. Marks does some basic molecular biology stuff. And then new this year, brand new, is Dr. Wei Li. Okay? He's brand new pharmacologist here, so he'll be teaching a few lectures. And you'll see him a whole lot more in the next block for pharmaceutical foundations. Okay? All right, I like to give everybody tips for success. Okay? Look, stay up with the material. All right? If you have a really good background in biology, chemistry, and biochemistry, all right, microbiology, as far as like your prerequisites, you just finished them, all right, and you have a really good handle on it. I'm one of the professors that, look, if you're bored in class and you don't want to come and you can do it on your own, be my guest, okay? But if you fail, don't complain to me, okay? All right? I'll just let you know that, okay? So one thing I can say is just be prepared, look at the lectures ahead of time, have any questions. Look, there's videos of all the lectures from all your classes, all right? I've heard students waste entire nights or drives re-listening to lectures if they would have just paid attention in class. I've had students who sit in class all day long and they do nothing. This is truth, okay? And then they say they go home at night and watch all the lectures again. You can guess that they weren't very successful, okay? I'll be honest, all right? At the end of the day, like with all your classes, hey, review what you learned. Stay up with the material. Inevitably, there's going to be some students who are going to wait for the last weekend to study for the block exam, okay? Now, some students can do that, all right? Be honest, there are some students that do that, okay? They're very good with that, all right? A lot of students in the past that I have to see after that first exam are not, okay? All right? And so just review the material, and so you keep staying up with the material every day, all right? Not everything's on the slides. I may make note of different things from time to time, so jot some notes down, okay? All right? You'll notice that I'll highlight things and outline things in red, okay? Those are probably important things, all right? All right? And ask questions. If you don't understand something, ask questions, all right? I try to define terms and things as I go, but if you are sitting there and you don't understand something, and if you're not going to Google it then, if you want to ask me, that's fine, okay? Don't get lost in lecture because there's some terms or some statements that you don't understand, okay? All right? I won't bite, I promise, okay? All right? Now, another thing. So if some of you guys are kind of, we have some non-traditional students who haven't been in class, they're not like right out of college or right from prerequisites and things like that. Look, if you need help, go get some tutoring here. There's a lot of good resources here at Campbell. I'm sure they've told you all about them in the orientation, right? Correct? Okay. So don't feel bad. Go get some help. Get, some pointers, things like that. One of the biggest things I'm sure they told you is time management and how to study. I can't tell you how many students, some who fail the first test, come to me and say, I study 10 hours a day. And then when you talk to them about how they're studying, they have phones, TVs, they're doing this, they're doing that. Look, it's quality over quantity, guys. Okay? I'll just tell you right now. Okay? And so if you don't follow these, you're going to be sad, and I'm going to be sad, especially after the first exam. Okay? All right? But if you follow all of them, you will win. Okay? All right? Any questions so far? If you guys looked at the course, I got 4 quizzes. Okay? Quizzes have anywhere from 18 to 20 questions. Pretty straightforward. I dropped the lowest grade. Okay? They're all announced. There's no unannounced quizzes. That's it. All right? You get the 3 block exams, and then you have 2 case presentations. Who likes teamwork? We got a few. Guess what? You got to do teamwork here. All right? So I'll put you guys into groups. You'll each get a question. There'll be a clinical case, and you'll have to do a little PowerPoint presentation on it. You'll each have to take a slide, and then as a group, you'll write it up, and you'll submit that. Okay? So, group work. All right? Look, it covers a lot of bases. A lot of it is review for the exams that we go through the cases, little points and things like that. The other thing is you have to learn to work together. Okay? All right, I'm sorry to say, there's always somebody doesn't like to work with, but, that always happens. All right. So, any questions about how the course is put together, what I'm expecting of you guys? Anything else? Nothing? They tell you, let me tell you about this right now. Okay, so, now, I'm not saying anybody's going to fail the first exam, but if you do, we have different levels of remediation. Did you guys read about that? They tell you about that? Okay, well, I'm not going to tell you. Because nobody's going to fail the first test, so I won't have to deal with it. All right? Right? Okay. All right, so we're going to look at basic chemistry and bonding. This should be basic chemistry you guys had as undergrads, all right, in organic chemistry and general chem. All right? Look back at it. There's a reason why I'm talking about all this stuff. Okay, and I'll try to point that out. Now, at the beginning of all lectures, pretty much everybody gives, at least that I know who gives them, they'll have objectives. All right? Now, all the questions that I'll ask on exams are based on these objectives. Okay? All right, just to let you know. And so, as you go through the lecture, things I point out, you look back at those objectives, hey, what's the information I need to know? Okay? All right? And if you can do all that, should be good thumbs up. All right? And so we're going to go through basic bonding. All right? Okay? What's the difference? Bless you. Covalent, non-covalent bonds. Right? Or ionic and covalent bonds. All right? Then, functional groups. I am going to ask you to know your functional groups. Okay, you guys should have learned those already, but go back and look through them. All right? And then classes of chemical reactions in cells. This is based on enzymes. So we're going to talk about a lot of enzymes later. Now, why do you guys care about all this? Well, I'll tell you. So, what are some different types of pharmaceuticals? And I've had to add to this over the years. Okay? So you got small molecules. What do I mean by small molecules? Are you guys really that tired, or are you just that scared? No, I'm just kidding. You can tell me. It's okay. Small molecules. Like what? Yeah, like what kind of drugs do I mean? Like, you got any examples? No? I'm sure some of you guys probably took some at some point today. Yes? Huh? Well, that would be over the counter. Yeah, so it would be a distinct chemical entity. NSAIDs. Anybody take some ibuprofen today? You get a headache? Aspirin? No? I guess, okay, well, so it was a really good day then. All right, awesome. All right. Now, what are some other big ones here? Peptides. Okay, you guys know what a peptide is? What's a peptide made out of? Here, say it louder. Amino acids, okay? So proteins. Can you name some protein pharmaceuticals? Here, say it louder. I can't hear you. Oh, yeah. Okay. Ozempic, yeah. Okay. Those are proteins, all right? Wegovy, all those ones. GLP agonists, okay? All right? We'll talk about those later on, and you guys will hear all about them in endocrine in the spring, okay? But we'll talk a little bit about it. How about antibodies? You guys watch TV at all? You guys got the Netflix with ads? Because that's what I got. The whatever, $8.99. You pay the extra? You pay the, oh man. Because they say with YouTube. Oh. Well, all the commercials, if you see them, it's like all these drugs, right? You ever see one for Humira, all these other immunomodulators? Those are all antibodies, okay? We'll talk a little bit about those later on, all right? So peptides are proteins. These ones, nucleic acids. You guys know what nucleic acid is? DNA, RNA, okay? All right, there are pharmaceuticals, approved pharmaceuticals out there that are DNA and RNA molecules. Can you name any? I know you've gotten vaccinated with some. The first COVID-19 one, yeah, from Pfizer, it was an mRNA, okay? All right, there are other drugs that are nucleic acid based, okay, because it'll be either an RNA or DNA backbone, okay, that modulates things like splicing, okay, or gene transcription, okay, thereby turning a gene off or on, all right? And then how about this one down here? You guys ever hear of cell and gene therapy? Any of those? CRISPR? You hear CRISPR? Yeah, and so these ones, a lot of therapies are based on taking certain immune cells out of your body, reprogramming them, growing them up, and pumping them back into you, which usually is certain cancer therapies. Okay? All right, gene therapies. All right, there's a number of them out there to fix certain genetic diseases. There's one of the big ones was sickle cell. A couple years ago, got approved. Okay, that's gene therapy. All right, they use certain viral vectors to carry certain proteins to make corrections within your own genes. All right, cool. And so we're going to be talking about all these basic processes, okay, to help you understand these mechanisms of action of all these different classes of pharmaceuticals. Okay? All right. 30 years ago, all you would have had to worry about is small molecules and a couple peptides. All right. But you guys, now, there's so many different types of drugs. Okay? Now, on to biochemical groups, all right, and linkages. All right, things like amines. This should be a review, guys. If not, go back, look it over, okay? Amines, you can have primary, secondary, tertiary. You can also have quaternary amines, okay? And that depends on the number of groups attached to that nitrogen, okay? These are important, all right? Now, I'm talking about all these, all right? Now, I'm making sure you guys remember all these because we're going to talk about them when we talk about other certain molecules and how these drugs interact, okay? And when you guys go on to pharmaceutical foundations, they're going to show you structures of drugs, okay? And you should be able to pick out what the functional groups are, okay? And what effect those functional groups are going to have on the stability of that molecule, okay? So we're going to talk about that basic stuff. and then they're going to talk about it in more detail. Okay? All right? Now, you have alcohols. You can have primary, secondary, or tertiary alcohols too, right? You should be able to pick those out in different molecules here. You have thiol groups, all right? It's sulfur, right? Sulfur, we'll talk about some different sulfur-containing compounds, all right? And amino acids, all right? Ether, and ether linkage, ROR. Anybody know where you find that? Where you find a lot of ether linkages? Well, how about ether and ester? Okay? So there's a lot of ether linkages in things like lipids. Because if you look at the difference between an ether and an ester, so an ether is, and R just means any other group, carbon most likely. Okay? And for the ester, you have, and then this is O, and then R. Okay? What do you notice that's different? You get that carbon with the double bond and the O and O. And so we'll start talking about, okay, let's see. Are those bonds up there, are those covalent or ionic that I just drew? Say louder. Covalent. Well, we'll get ahead of ourselves. What's the difference between a covalent bond and an ionic bond? What's that? Covalent, I heard shared. Here, somebody raise a hand. I can't.
One is easily broken and the other one isn't?
No, they're actually both pretty strong. But think about the charge. Okay? All right? Something, and so have you guys heard of electronegativity? Remember electronegativity? Okay? Some atoms, what? They really want extra electrons, right? Okay? Electronegative. All right? And other ones don't. Okay? And you look at the periodic table, we'll talk about electronegativity difference. And if they're really far apart in their electronegativities, basically group 1 and group 7, okay, they're going to form what's called an ionic bond because their electronegativity difference is so great. Okay? All right? But a carbon-carbon bond, carbon and carbon, right? That would be considered a pure covalent bond because there's no difference in the electronegativities. How about carbon and oxygen? There's a difference, okay? And what we're doing is introducing, when you see these things like this, when you're looking at it, there's going to be partial charges, okay? Or dipoles. It's going to be a polar molecule, okay? And that's going to affect its stability. We'll talk more about it, okay? So anyway, an ether, because of the way the bonds are, is much more stable bond than an ester. All right, how about an aldehyde and a ketone? Yes?
Can you repeat the difference between covalent and ionic bonds?
Well, we're going to go in more detail later on. Okay, you're welcome. All right, aldehyde and ketone. Okay, in sugars, you find aldehydes and ketones. An aldehyde is what? One at the end where you have the hydrogen. Okay. And a ketone is an internal carbon-oxygen double bond, right? Okay? And we'll talk about that more when we talk about sugars later on. All right, carboxylic acids. All right, acidic groups, okay, important for ionization. All right, thioesters. Basically, it's a ester there, but instead of O, you got an S. All right, sulfur, okay? This is important for certain vitamins, okay, which carry around acyl groups. An acyl group is just a 2-carbon unit for fatty acid oxidation or fatty acid synthesis, okay, and some other processes, all right, in the TCA cycle. We'll talk about it later, but there's a reason why I talk about all this, okay? Some other ones, amides, amide bonds. Where are you going to find an amide bond? What type of pharmaceutical? Primarily. What's that? Peptides, amide bonds, okay? From amino acid to amino acid, all right? Amines. These are one special ones, okay? But you'll find these in a lot of small molecules. All right, disulfide bonds. All right, this is disulfide bonds between two cysteine amino acids. You guys heard of cysteine? Talk about, so it has a thiol group, and it'll form disulfide bonds, a covalent bond, okay, that helps stabilize protein structure. Okay, all right. There's a bunch of these disulfide bonds into certain antibodies. Okay, a phosphate ester. Okay, okay, or basically this phosphoryl group here, phosphate ester. Okay, all right. In a cell, okay, there are a number of proteins that are regulated by phosphorylation. You guys ever learn that? Okay, so yeah, so certain proteins are activated by phosphorylation, okay, and then some are, inactivated by dephosphorylation. Okay, all right, and so this will be things you'll see attached to serine, threonine, and tyrosine. Okay, all right, and why? Because they have a carbon OH. We'll look at it later. All right, they have an OH group on their R group. We'll talk about that. Okay. Now, how about a diphosphate ester? Okay, or a phosphoanhydride bond. Where are you going to find those? ATP, yes, ATP. Okay, and this is where all that energy is stored. Okay, so ADP, ATP. How about a phosphate diester? Okay, or a phosphodiester bond. Where are you going to find those linkages? Nucleic, say it loud. Yeah, nucleic acid. You guys say it louder. It's a big room. So it's a backbone of nucleic acids. All right. All right. So alkanes, just go through it. Alkenes, double bond, right? And so these are important. We'll talk about these later. When you go from a single bond to a double bond, what happens between that bond? So a single bond, you can still get rotation. Okay, double bond, you can't. So double bond, you have to start, well, we'll be thinking about it when we talk about proteins. You have things like cis or trans, if my thumb were in the same group. Okay, and so that has implications for the structure. All right, alkynes, triple bond. Okay, then we have different groups. We have aryl groups. Okay, and then these are specific examples of phenol. So, or they call this a phenyl group too. Okay, but a phenol, OL, alcohol, right? Okay, OL. All right. Now, you can also have heterocyclic rings. Okay, an example of one of those. Anybody know what this molecule is? All taken it at some point in your life, I'm sure, unless you're allergic to it. Or you probably took it once then. Yes? It's penicillin, yeah. Okay? So this is penicillin. It's going to, all right? And so when you have rings with a carbon and another type of atom, this one's nitrogen, this is a beta-lactam ring, that's what it's called. And this is the part that actually interacts in the chemistry with the excuse me, transpeptidase in the bacteria. All right. All right. Ortho, meta, para. Any questions? Yes, no, maybe. Just nomenclature, guys. Now, halogenated hydrocarbons. Why am I going to talk about the halogens? And that's this group here. Okay? So, what do you guys know about those? They're really electro what? Negative, yes. Okay? And so they're going to have specific properties. What else? As you go down a group in the periodic table, what happens to those atoms? Radius increase. Okay, they get bigger, right? Because of the bigger nucleus, okay, and the more shells. Good. Okay? Well, the reason I'm bringing it up is there are halogenated pharmaceuticals. Okay? And there's a reason for this. They make them more stable, okay? So it can affect their pharmacokinetics, right? You guys know what pharmacokinetics is? What happens to that drug after it gets into your body, how it gets excreted, all right, how your body changes it. We'll talk about a lot of that. And then, okay, and so these can have effects of making it more stable, giving it a longer half-life in your body, and or for better absorption. Okay? This one is a non-nucleotide reverse transcriptase inhibitor. You'll notice it's got chloride and it's got fluorines there. Okay? All right. Now, those bonds with those really great, I mean, it's ionic, it's almost ionic. Okay? All right, because of the big electronegativity, it's really stable. Okay? And it really increases the half-life. All right? Then we got antihistamine. It's bromolylated. Okay? And then this one. You guys should probably recognize that one. All right? Thyroxine. All right? That's natural, but they also make it, you know, our body naturally makes this, but it's also made this way. Okay? There are some other ones. For instance, there's some other nucleotide analogs with bromine or iodine attached to them. And what happens is in bacteria, they kill bacteria they get incorporated into the DNA, and you have these big atoms, all right? And it distorts the DNA, and it ends up getting digested and messes up the bacteria, okay? So, all right. More functional groups you might see. An amide, all right? Urea. We'll talk about urea when we talk about the urea cycle, okay? All right. Carbamates, all right? This one, I don't know how much you see that. Guanidine, you're not going to see that very often. But some of these ones you're going to see in different small molecules, okay, they're going to talk about, all right? Sulfoxides, sulfoxones, and sulfonamides. There are a number of pharmaceuticals with sulfonamides in them, okay? All right? And then this straight from methyl, single carbon, ethyl groups. You got a phenyl or aryl, okay, group. All right, and this one, imidazole. Is that a heterocyclic? Yes, got carbon and nitrogen in the ring, okay? So it's heterocyclic. This is found as the side chain of histidine, and it's an important one we'll talk about later, okay? Now, one of the things, all right, those electronegativity differences we're talking about, it's going to help stabilize, destabilize, make things polar, part of that molecule polar, okay? All right, and that gives, you know, if you have certain charges, that's going to give certain properties to that molecule, right? Okay, as opposed to just being a completely, you know, C18, all right, carbon chain, all right, a lipid, okay? All right, now, the reason I show you this, and I'll ask you some questions on it with these functional groups, when you guys go to pharmaceutical foundations, the first thing that Dr. Howe makes you do is take a test on nomenclature and recognizing functional groups. So you guys should all be gangbusters after doing this, okay? All right, and these are the kind of questions. So, you know, if you look at a molecule here, this is histidine, the amino acid, okay? Has an imidazole group, primary amine, and a carboxylic acid, okay? Look at epinephrine. All right, what are some of the other ones here, okay? Your amino group. You could say aryl group. You could say a phenyl group here, alcohol, all right, etc. Okay? All right, I want you guys to look at that. Now, some more terminology. You guys heard of hydrophilic, water-loving, hydrophobic, water-hating, okay? Other people might even say, instead of hydrophobic, they say lipophobic, okay? Hydrophilic, lipophobic, okay? Same thing, all right? Depending on what you're doing. Now, talk about this later on, amphipathic, okay? A lot of molecules are amphipathic. They have both hydrophilic groups and hydrophobic groups. Okay? That's usually important for their function. All right? Nomenclature, guys. Now, what's the name of the functional group shown here? You guys tell me. Is it a sulfhydryl? No, there's no sulfur anywhere. How about a diene? No. Alcohol? Carbonyl? Yes. Okay, good. Excellent. It's carbonyl. It's not an amine. All right. Let's see. What's the name of a compound with this functional group? What's the name of the compound with the functional group? Remember the sugars I was telling you about? You guys ever seen sugars, like especially the Fisher diagrams? You know what I'm talking about? Where you have these are the carbons, where you'd have a double bond O, and you have another one up here. What's it called? Is it an ether? No, ether's this, right? Aldehyde? Aldehyde, what would this have to be for it to be an aldehyde? Hydrogen, good. Ketone? Yes, ketone, okay. An ester, what would this have to be an ester? An O, good. All right. Good job, guys. All right, does this compound contain an aldehyde or a ketone? All right, what's this group? Carboxylic acid. Okay. What's this group? Alcohol. Okay. What's this group? Aldehyde. Good. Okay, so it's an aldehyde. Now, like we just said, though, if it was internal, okay, then it would be a ketone, right? All right, good. Oh, we're skipping that. Now, bonding and stability. Okay, this is where we're talking about the difference between ionic and covalent bonds. Okay, and the real difference is, okay, a covalent bond, you're sharing electrons, right, guys? Okay. Now, this has to deal with, okay, they're going to share electrons. Okay. Now, an ionic bond is when that electronegativity difference between these two atoms is so great that one takes that electron and the other one gives it up. Okay? And so therefore they have a what? Negative and positive charge on them then. Okay? All right? So completely electrostatic interaction. All right, it's very strong. Okay? Now, all the other ones though will form covalent bonds. Now, you can have everything from a carbon-carbon bond, right? Covalent bond. That would be considered a pure covalent bond because there's no difference between the electronegativity between those two atoms. You guys follow me? Okay? And so once you go to different ones, okay, oxygen, what's going to happen? There's going to be a partial charge here. What's the partial charge on the carbon going to be? Huh? I heard it. Because oxygen is more electronegative. Those electrons would like to hang out with it more than with the carbon. And so there's that. It becomes a polar bond, right? Polar. Okay? All right, good. Now, electronegativities, just remember, all right, they increase going this way and this way, with fluorine being your greatest there. Okay? All right? And here were just some examples of ones here. It's just showing you, we talked about it. It's the difference between pure covalent, covalent, and ionic. You guys got that? Okay, good. Now, carbon-carbon bonding geometry. Okay? All right, this is what I was talking about when you have a single bond. All right, single bond can rotate. Okay? That molecule is going to have a lot more, what? Spaces that it can occupy, and so the structure is going to vary a little bit, right? Okay? What happens when you have a double bond? Okay? You have a double bond, it gets locked up, and so you end up with this thing called cis and trans. Okay, we'll talk about. Okay, all right, they can't rotate anymore. All right, this also happens in amide bonds in the backbone of proteins. Okay, it's not a double bond in that amide bond, but because of those electronegativity differences, it acts like a double bond, and so there's no rotation. Okay, all right. Okay. Now, the covalent bond strength, you can imagine, all right, relative electronegativities of the bonding atoms. It's going to deal with that, all right? The distance of the bonding electrons from each nucleus, right? Okay? Two really small ones are going to be much stronger than, say, two really big atoms, okay? The distance, okay? The nuclear charge on each atom, all right, how many protons it's got. All right, and the number of electrons shared, triple, double, or single, okay? Pairs, all right? And this was just to show you guys, and I'm sure you guys all remember this, okay? Just to show you guys, don't memorize any of this stuff. Single bonds compared to double bonds or triple bonds, the amount of energy needed to break them, okay? It takes a lot of energy to break those triple bonds, okay? All right, as opposed to the alkanes, all right? Single bonds. All right, cool. Now, more important stuff, at least in my opinion, all right? Dipole moments, okay? Now, just because some molecule has no net charge, okay, there can be an asymmetric distribution of the internal charge, okay, because of these electronegativity differences. So it's not, it's not a pure covalent bond, all right? And so we'd say these molecules are what? Polar, okay? Depending on the molecule and the structure of it, it can have a permanent dipole moment. You guys heard of this? Yes? No? Maybe? Okay? And if you look at some of these, so they'll have these dipole vectors, and this is the overall one, okay, for showing up here. Now, I always have some students tell me, I learned it like this, okay? All right? Or the arrow is the same thing, all right? The arrow is the positive side, all right, over here, all right? And it's just showing you that what? There's a slight positive charge here and a slight negative charge here, okay? All right? Carbon dioxide, special circumstance, okay? You have the partial charges, okay? But when you look at the dipole moment, you guys have to think back to, I guess they do it in calculus and algebra. You guys learn vectors, how to add two vectors together. That's all the vector sum of the dipole moment's doing. You're taking those two vectors. Well, what happens when you have two opposing ones? They cancel each other out, and so the overall dipole moment is 0 for that molecule. Okay? Water, okay? Shown overall there. Now, these are ones the same way. So we have what here? If this is our number 1, what position is this in? Ortho, okay? So ortho, all right? Ortho, or meta, all right, can actually cause an overall dipole moment in there. But look what happens when you have something at para. Same thing as carbon dioxide. It crosses itself out. Okay? And then when you're looking at this over the course of this whole molecule, like glycine, it's a small amino acid. All right? Dipole moment's pretty large. Okay? All right? So anyway, these have dramatic effects. Why am I talking about it so much? Why should I care about why should you care about this when all you got to learn is what how drugs work? Well, when you take a pill, how does that drug get into your body? Yes?
Wouldn't this affect like the absorption rate of the medication?
Yeah, so absorption. It all has to do with what? The charge on your drugs when they're in certain compartments of your body. Okay? If something's really highly charged, do you think it's going to cross a membrane? from your cells? Huh? No, it's not going to, all right? Unless it's actively transported, okay? It's not going to do it, okay? And so all these groups, all their properties, it all depends on what happens to those different groups in those molecules for the pharmacokinetics, okay? Now, all these different polar molecules, all right, dipoles, all right, they then can also interact. So between two different molecules, okay, intra or intermolecular, all right, and these would be called non-covalent interactions, all right. And so some key things, all right, from these polar molecules interacting, polar and polar, okay, is it's much weaker than a covalent bond, okay. Now, a lot of these interactions that happen are weak. Now, that's a good thing, okay? When two things bind in your cell, if everything formed covalent bonds, okay, we probably wouldn't be alive, okay, if it's that stable, all right? And so they're transients, okay? And so that's important for binding, all right? And so these things have to bind, release, bind, okay? And so they have to do that. And most of the things that we're going to talk about, it's all electrostatics, all right? Positive and negative things getting together, okay? All right, for a lot of different drugs and different pathways that are going on, okay? So now, these dipole moments can interact, okay? And so if you have two permanent dipoles, okay, they can interact. All right? These are permanent dipoles, okay? Now, you can also have a permanent dipole induce a dipole or polarity in another group. Okay? So you can have these. All right? And then you can have these things called London dispersion forces. You guys ever hear this? Induced dipoles, okay, because of atoms. All right? And it's very weak. But when you add all those up, say, in a protein structure, it makes it very stable. Okay? Now, the only thing I want you guys to get out of this, okay, there are different types of non-covalent interactions. Now, these ones, charge, you guys can guess, okay? All right? Two specifically charged groups, okay? All right, it's like an ionic bond almost. All right, but charge. All right, charge dipole. Okay, all right, dipole. Charge-induced dipole-induced dipole. Dispersion, that's London dispersion. And then we have hydrogen bonds. Now, over here, what I really want to look at is, okay, dependence of energy on distance, okay? Now, what this means is this is 1 over r, the radius between the two atoms, okay? Now, the energy, it's relatively stable, 1 over r, okay? Which means it can be relatively far apart, okay? Now, as you go down all the way to these dispersion, induced, it's 1 over r to the 6th, okay? And basically what it's saying is they have to be really, really close for that interaction to be fruitful, okay? All right? So it's just distance-wise, all right? Now, our lone guy out down here, Mr. Hydrogen Bond, okay? You guys heard of hydrogen bonds? Very important, okay? Makes things soluble, very important proteins, everything, okay? Now, what makes them so interesting is they have a fixed length. Okay? All right? As opposed to these other ones, they can still have interactions and be farther apart, especially the charge, or the charged dipole. They could be in a range of distances. Hydrogen bonds have a specific length to them. Okay? Now, this has implications for things like proteins and protein structure and nucleic acids, okay? Giving them an exact structure. Now, you guys ever see hydrogen bond here? You guys all heard of hydrogen bonds? Yes? Okay. You have a donor, okay? And you have your acceptor, your negative, okay? All right, so you have your positive and negative, basically, right? Okay. All right, and it's a fixed length bond. Okay. I thought I talked about. Oh wait, I thought I had another thing on there. I guess I didn't. I talk about it later when we talk about solubility. But as far as your hydrogen bond here with the fixed length, now these can either be, okay, intramolecular in a molecule or intermolecular. What do I mean by intra and inter? I'll go back to nomenclature here because I'll talk about this a lot. Intra is within the same molecule. Intermolecular is between two different molecules. Okay? You guys good with that? If you guys terminology, just say, hey, stop. Enzyme classification system. I only bring this up because you guys are going to talk about a lot of them. Okay? And when we talk about enzymes, all right, this is more for biochemistry. There are 6 types of general reactions. And all the enzymes that are out there, and some of them are used therapeutically, okay, they'll be, the first number is this, what type of reaction they catalyze, okay? The reason I put it out there is, number 1, oxidation reduction reactions, okay? This is one of the most common ones you guys will learn about when you talk about cytochrome P450s. You guys heard of those? In your liver? They're one of the first things to detoxify xenobiotics from your body. Okay? All right? Anyway, the second one, group transfer reactions. Those are really common too. Also in drug metabolism, but also in covalent regulation of other proteins by what? Group transfer, like a what? Phosphate. Okay? So you phosphorylate a protein at a certain site, you activate it, etc. Okay? And then this one I'll just mention, addition or removal of water. Okay? So basically a hydrolase. All right? Either you join something together and lose the water, or you add a water and break something else. Okay? Now, to give you guys a difference, so this here is the same nomenclature as this. Okay? It's the same enzyme, sorry. This is from the enzyme, and this will have what reaction it catalyzes, a subgroup, and some more specifics, okay? All right? Now, this one over here is more the genetic. What happened? Thing stopped. Is the genetic classification here. And so CYP is the gene, cytochrome P450. Okay? You guys ever hear those? Yes or no? People saying yes? Okay, good. Now, this are the different subtypes of it, okay? All right, as far as the genetics of it, okay? And so CYP is that. 2 is the family. This is a subgroup, okay? And then they'll have the gene. And then you'll see other things by the different alleles, allelic variants. Now, this is just to introduce something to you called pharmacogenomics. Anybody hear pharmacogenomics? Yes? No? No? Okay, well, is everybody in here identical? No. Okay, we all have variations in genes, all right, mutations, things like that. Well, we have it in a lot of these different genes that help in the elimination of certain pharmaceuticals, small molecules. Okay? Now, pharmacogenomics deals with the variation in the population. Imagine if you have a mutation in this cytochrome, it's responsible for eliminating drug A. Okay? All right? But if you have this certain variant of it, you don't get rid of that drug as fast. What's that going to mean for you?
Toxicity.
You could get really too high with it, yeah, and it could be toxic. All right? Okay? And so a lot of different drugs are They're like that, okay? You guys ever hear of warfarin? Yes, everybody heard of warfarin, okay? Blood thinner, okay? Stops your blood from clotting. There are certain genes, okay, where they can look at the certain mutations that you have there and figure out exactly what dose they should put you on, okay, as they monitor your clotting later on. Okay? Otherwise, it could be really dangerous, and they could just bleed to death. Okay? All right. I got a couple other questions here. Oh, oxidation reduction. Forgot about it. You guys know what oxidation reduction is? Forgot. Oxidation? Reduction? Oxidation. What's oxidation? What's the most oxidized molecule up here? What's the most oxidized molecule shown on there? What's that? No. What's that? No.
Carbon dioxide,
What's that?
dioxide.
Here. So carbon dioxide, okay, right? It's the most oxidized, all right? Okay? Now, why am I bringing this up? Okay, we're going to talk about oxidation reduction reactions a lot, okay? I have that. And so when we talk about metabolism, all right, I'll kind of break it down into anabolic and catabolic. Anabolic, building things up, right? Catabolic, breaking them down, okay? And in general, all the anabolic processes are doing what? They're reducing things, okay? All right, adding more energy, all right, by, all right, that gain of electrons, all right, or more hydrogens there, okay? All right, versus catabolism, okay, breaking things down like in our body, all right, we're ending up with what? The most oxidized thing. We're taking all that energy out of it, okay? We'll talk more about it, all right? So go back, look at oxidation reduction. Should be basic, okay? And then I told you about the other one. What? This is the, what? EC what? Number 2, group transfer. Things like, phosphates, okay? Phosphates to proteins, phosphates from ADP to ATP in the cell. All right, another one up there I got up there for glucose, okay? Where you phosphorylate glucose. That's the first step of glycolysis, okay? When glucose comes into your cell, all right? Why do you think it charges it with a phosphate? Why do you think it makes that molecule? There's a couple reasons. One is, what's that phosphate have on it? Lots of what charge? Negative, okay? That's going to keep it locked in the cell, first off, okay? But also, you end up with what? That phosphate bond, that carries energy, okay? It's a good leaving group because it's so electronegative, okay? The big that big dipole moment between that molecule, okay? All right? And you'll see that if you guys, so when you guys did microbiology, you guys do any metabolism? Yes? And they talk about good leaving groups and things like that, where you have to activate a molecule with a phosphate or some other type of group. Okay? Well, this is the same thing with, we'll talk more about it later. Get through this. Condensation and hydrolysis. This is what? EC number 3, right? So you're using water. All right? You're either, what? Losing a water or adding a water to break it down. Okay? All right? Now, is this compound hydrophilic? Yes. Why? What can all these oxygens do? And nitrogen. Exactly. Okay? Well, we're going to talk more about that. But basically, things that are going to form a lot of hydrogen bonds are going to be really soluble in aqueous. Good. Which of the following molecules would you predict to have the greatest solubility in water? Who wants to take it? Go ahead.
C because it has the benzene ring, so a lot of hydrogen,
So it has a lot, but this group, okay, with the carbon and the hydrogen, okay, that carbon there, all right, you can't have hydrogen bonds, okay? All right, that's a very, actually, lipophilic group because of all the carbons, okay, in that ring. Yes? Go louder. It has at least the aliphatic character. You guys hear aliphatic? Just means the degree, number of carbons in it. Okay? Carbon-carbon bonds. And so, yes, it's going to be this because they all have what? They all have that same group. Okay? All right? Cool. So you have to look at the whole molecule. All right. Which atom does not commonly form hydrogen bonds between or within biological molecules? I think I already. Which one? Huh? Carbon. When you look at the donor and acceptor, okay? All right? Carbon. Okay? We'll look more at it later. All right. Guess what? You guys need to take a break. Take a break, and we'll come back. We'll talk about pH, basically. You guys good with pH? We'll skip it. We'll skip it, and you guys can just ask me some questions tomorrow. Yes? No? No takers? Okay. All right, guys. We'll get finished. I'll get you guys out of here. You guys can go take a nap and then go study, right? You're going to study, right? Yes? Just lie to me right now. That's okay. It's all right. All right. So water's acids and bases. All right. Look, most of this should be pretty straightforward. The couple terms that I'll start talking about a lot is Ka, dissociation constant, and pKa. Okay? All right? And we'll talk a lot about pKa towards the end. And you guys will talk even more about it, okay, when you talk about the pharmaceutical foundations course. Okay? All right? Anyway, we'll talk about that. Now, water. What do you guys know about water? Okay? What? Everything's aqueous environment, right? Okay, carries everything, helps out with reactions, okay? Forms hydrogen bonds, etc. All right? Now, H bonds. Remember I said they have a fixed length, okay? Now, with that fixed length, all right, you can have ones that are either linear or bent. But the length, okay, the length on these, depending on how they're arranged, okay, all right, is always the same, almost, okay? Any diehard chemists? No, good, okay. That's a long conversation. All right, so with that, all right, so you can see this is in line with it. This is, okay, so weaker, all right? Now, with those, though, and they have that precise distance, okay, all right, it gives those molecules, all right, hydrogen bonding within itself, intramolecular, okay, it gives it a three-dimensional structure, okay? All right, structure, when we talk about different, whatever it might be, binding partners, being enzymes, receptors, etc., okay, Structure equals function. Okay? Now, hydrogen bonding. Okay? For hydrogen bond donors and acceptors. All right? What makes something soluble? It's going to hydrogen bond with water, right? If we're talking about something that's aqueous. Okay? All right? And so that's all I had up here. There was just some, differences here. This is in the backbone of what? Peptides. Okay? All right, this would be intramolecular hydrogen bonds, or it could be intermolecular, depending on what it is. And this, you'll know that A, when they say A binds to T in DNA and G to C, all right, A and T, there's 2 hydrogen bonds that link it up, right? And G and C has 3. Okay, good. All right, good. And these are sugars, different sugars here. This would be a what? Ketone, right? Hydrogen bonding. Okay, so it'd be a keto sugar. All right. Bond energies. This is just the same thing again. Look, covalent, very strong. Okay. Our non-covalent ones. This is the only point I'm going to make. All right. And you'll see it goes down. All right. You got your ionic and van der Waals. Hydrogen bonds are right in the mix there. Okay. So they have some strength, but they're not super stable like a covalent bond. Okay. All right. Now, when we talk about enzymes later on, when you think about something that's going to inhibit an enzyme, okay, all right, some drug that's out there, some small molecule, some of them are even called suicide inhibitors, okay, because they form a pseudo-covalent bond with the active site, okay? All right, it takes a while for it to dissociate, okay? This happens with certain toxic chemicals, too. I'll mention later on, all right? So, solvation. What's solvation? They'll talk about this a lot in pharmaceutics. All right? And basically, it's solubility of the molecule depends on the ability of that solvent to interact more strongly with the solute than with each other. What's that mean? So, whatever you throw into, say, a glass of water, all right, if that water is going to want to interact with what you threw in more strongly than itself, it's going to dissolve it, right? It's going to solvate it, okay, and dissolve that molecule, all right? But say it doesn't, like a lipid, okay, something that's hydrophobic, okay, it's not going to work as well, all right? Now, so with that, with the solvation, all right, this is a hard concept. You guys remember thermodynamics? Delta G? You see some heads? I'll bring it up a few times. I'm sorry, I have to. Delta G equals what? Delta H? What's H? Enthalpy. That's the heat of the reaction given off, okay? And then what's delta S? What's S? Entropy. What's entropy? One to more disorder. So now if I said, like class is over right now, get out of here, and you guys run out, what's going to happen? Entropy is going to go through the roof, right? Okay, because you guys are moving. Right now it's really low and everybody's half asleep. Okay? All right? Well, same thing for any kind of molecules, okay? Well, if you think about some, really long molecule, okay? Long spread out molecule, okay? It's about surface area, okay? There has to be a whole bunch of, say it's all spread out like this. And it's not real hydrophilic, okay? But the water, what's the water have to do with that in there? It still has to kind of isolate it, okay? So it still pseudo-interacts with that hydrophobic molecule, right? Okay, so it coats it, all right? Got me on this? And so if you think of the surface area between, say, like an elephant and a mouse, okay? All right, large surface area, small surface area, okay? All right, compared to its mass. How do I do this? This is always hard for me. So you have to have a whole bunch of water molecules lined up around it. Well, if that thing then folds up into a nice compact ball, what happens? Not as many, and it doesn't have as much surface area, okay? What happens? Less water molecules have to isolate that. Okay? So actually, entropy increases. Okay? So if you have something with a really, really, really large surface area, a lot of water molecules have to give up all their freedom and sit here in class and listen to me. Okay? Now, if they all fold up into, really tight ball, okay, only those ones on the outside have to give up their freedom. And everybody else can still jump around. Okay? And so it's entropy that drives this process. Okay? And this is called the hydrophobic effect. Okay? And this is when we talk about proteins. This is what drives proteins to fold into their native structures. They give them that native structure that gives them that function. Okay? Now, pH scale. You guys know the pH scale? Measures the hydrogen ion concentration. Yes? No? I don't want to harp on it. Yes? Okay. Well, I'm not going to go on some of these. Now, this is just some examples of, the pH of some different solutions. pH of our blood actually is like pH 7.34-ish. Okay? It's tightly regulated. All right? And we have our, what? Gastric acid, hydrogen chloride, right? Yes? No? Okay, really acidic. All right, and then you can go up here to bleach, all right, or ammonia. All right, they're going to have what? Higher pH, okay? Or a lower concentration of what? Hydronium ions, right? Okay, good. All right, just have that in your mind. Now, this is some term you'll see. They'll use it in different aspects for receptor binding or the dissociation of acids and bases. Okay? Now, it's called Ka, dissociation constant. And so if you look at an acid in water, what's it going to do? It's going to become what? This is the acid. What's this molecule called? The conjugate what? Base, right? Okay? Conjugate base. All right? What's this? Actually, no, this is our conjugate base. Sorry. I hate when they do that. Just H+. Okay? It's our hydrogen ion. It's our conjugate base. All right? Because why is it a base? What's the difference between an acid and base? Basic definition in biological conditions. What's an acid do? What can an acid do?
Donate protons.
Say louder.
Donate protons.
Donate protons or hydrogens. Yeah. What do bases do? Suck them up. Good. All right. Awesome. Got it. All right. Now, and so this Ka is what? It's going to be the difference between these species. Okay? All right? The dissociation of this into hydrogen ions at the conjugate base. All right? You guys following me? All right? Now, they'll use this as a way to measure the strength of an acid. Okay? Now, if it's a really strong acid, like HCl, right? Okay? That has a Ka that's much greater than 1. All right? What's that mean for this ratio up here? It's almost all going to be what? It's going to be almost all that. All right? Completely dissociated. Okay? The strength, that Ka just says, hey, I'm going to give up that hydrogen really easily. All right? The higher that Ka is. Now, this doesn't really work, though, for weak acids, okay? Really weak acids. I'm talking really weak acids for us, okay? That we're going to talk about in class. And so, you guys heard of this equation? You guys had to in chemistry class. Yes? You know how to use it? You remember how to use it? They're going to make you in pharmaceutical. I'm not going to make you calculate anything. It's okay. All right. But what does this do? And so really it relates the pKa to what? The log. So it's basically what's ionized and what's not. How much is dissociated and how much isn't from the acid to the conjugate base. Okay? All right? Now, and you guys will have to use this for certain molecules with certain side groups, and they'll be like, hey, what percent of this drug is ionized in the stomach? So you'll know the pH, you know the pKa, and so you've got to figure out the ionization. Okay? I'm just telling you, that's what they make you do. Okay? All right. Now, the great thing about this, okay, this is where things come in, this pKa, okay? Now, how is this? So this is saying what? What's that ratio going to be? Conjugate base to, what is it? I heard it. 1, right? So that means they're equal. 50% of the acid is in the acid form. 50% is in the conjugate base form. Okay? All right? And when that happens, that's pH equals to pKa. Okay? All right? Now this is for an acid. Remember, an acid. Okay? And so, this is important. Good cheat sheet for extent of ionization. I'll go through this a couple more times today because students always have the worst problem with this. All right? When the pH equals the pKa, it's what? 50% ionized, right? 50-50. Yes? Okay. Now, for an acid, okay? When the pH of the solution that it's in is the pKa plus 1, okay? Give you guys an example of a weak one, a carboxylic acid in some protein, okay? All right? The pKa of this will be roughly, we're just going to use 4, okay? So at pH 4, what's that going to mean? That group is going to be what? 50% ionized. 50% of it's going to be ionized, 50% isn't, okay? Now, if we go to pH 5, what's going to happen? The higher pH you go to. Remember, this pKa, all right, is just that, like the Ka, is just a propensity of that acidic group to give up the hydrogen. Okay? All right? So when the pKa, when the pH equals the pKa plus 1, so I'm saying it's pH 5 now. How much of that? And it's right here. So it's going to be much more likely to give up its hydrogen. Okay? What happens when you go to a higher pH? What does higher pH mean in terms of concentration of hydrogen ions that are there? It's lower, right? And so that acid, as you get farther away from the pKa, higher above it, there's less hydrogen ions around. It's going to give up more and more likely, more quickly. Okay? So as you get farther above the pKa, what happens? All right? Even at plus 2, all right, at 2 pH units above, it's almost 100% ionized. Okay? It's a little cheat sheet. Now, bases. This is where it gets tough. Okay? All right. So, when we talk about different groups we have, all right, so in addition to having things, acids, straight acids like carboxylic acids or other groups, we also have what? In all our proteins and a lot of things. A lot of amines, right? Okay? Now, ammonia. Is that an acid or a base? It's a base. But we're talking about pKa, right? Not pKb. Okay? And so that's why, if you look at this, if you look at ammonia, all right, plus to, what is this? No charge, right? Okay? So what do you have? So same thing. When pH equals pKa, it's going to be the same, okay? Now, when a base, Okay, if you have a base, all right, what does it become? Huh? Well, just the opposite. Think about the other. So we have an acid becomes a conjugate base plus a hydrogen ion, right? We have a base becomes a what? Conjugate acid, not conjugate base. Okay? And so what this is looking at, all right, is that ammonia group. And so this is the pKa minus 1. Okay? All right? Minus 1. Ugh. You guys are going to have trouble with this. I know you are. Some of you will. All right? Because people get confused. And so the farther you go below, okay, it gives up that hydrogen. Okay? The conjugate acid does. Okay? We're talking about the conjugate acid of it. Okay? All right. We'll talk more about it. All right? But because we have to talk about buffers. All right? You guys, most students are like, why do I need to know about buffers? Okay? All right? Any of you guys work in a hospital on any things? Do you ever have to help with making IV bags? Any of the people? Yeah, I see some head. Why is it important? Why is buffers important? Well, think about what we talked about. So, different types of drugs. They have different types of groups, right? Functional groups. Okay? Some are more hydrophilic or hydrophobic. Okay? Some form more easily dissolved. Okay? Depending on what? The pH, right? Especially if they're ionized. Okay? And so, if you think about if you have to prepare an IV bag with a certain drug, and it has a pKa the same as the pH of your IV bag, what's going to happen? It's not going to be very soluble. All right, that's when it's least soluble. We'll talk more about that later, and it's going to fall out of solution. Okay? All right, so solubility matters for all this stuff. All right? And so have you guys ever heard of, different, when you have buffers that, you want to be near the pKa? A buffer is good at a pH equal to the pKa and then plus minus 1. All right? And that all has to do with the fact that if you go minus 1 or plus 1, it's already 90% ionized. And what's a buffer going to do? This is just an example of titration curves. Okay? And so this is 50%. This is the dissociation here. Okay? And across here is the pH. And so where there's 50-50, that's what? That's the pKa, right? Yes? You guys still remember that? Okay? And so as you go up it, what happens? It gives off the hydrogen, and you end up with more of the conjugate base, more ionized molecule there, right? You guys know what I mean by what ionized means in general? What? Somebody? I heard it. Say it louder. Please. Say it. Somebody said it. I think I heard it. Something that's charged, okay? In case you guys, all right? Something that's charged. Okay, and so the farther you go up, what do you have here? NH4 plus. That's actually the conjugate what? Base. Okay? And so anyway, you're going to, why is it the conjugate base? What do acids do? What do acids do? Give up protons, and it has a proton to give, okay? So conjugate base, all right? Okay, and so as you go up, you give off that hydrogen. As you go down, you gain one, okay? And it's the ability of that acid to either take up or give off hydrogens that keeps it buffered near its pKa, okay? This is just an example with acetic acid, okay? As you add in hydrogen, what's going to happen? The conjugate base is going to take that hydrogen and make acetic acid. What happens if you throw in a base, sodium hydroxide? What's it going to do? Well, that's going to dissociate to the conjugate base, and you're going to have hydrogen plus OH give you water. It's going to keep the pH the same, right? It's all about the concentration of hydronium ions. Yes?
So when you're saying that the pKa goes down,
No. pKa doesn't go down. pKa is a distinctive characteristic of that acid, or yeah, that acid. But as pH changes, because pH is what? Just changes the concentration of hydronium ions. All right? Okay. Now, another example of this. Breathing, our lungs, okay? You guys know what this is? Carbonic acid. You guys know what this is? Yes, say it. No, think something else. All right. Chloride. No chloride. Bicarbonate? You guys hear of bicarbonate? Yes? No? So this is one of the main buffering systems in our blood. Okay? And so what do we have here? If we have too much acid coming around, right? Okay? It will what? Link with the conjugate base of carbonic acid and form carbonic acid. Carbonic acid then does what? It actually gets dissociated. It breaks down, okay, CO2 and water. Carbonic anhydrase. It's an enzyme. And this is happening in our red blood cells, okay? All right? We're getting this stuff done in our red blood cells and in our blood, okay? All right? And so what's going to happen? So we're getting rid of hydrogen ions, okay? So we're going to do what to our pH? pH is going to do what? It's going to go up, right? If we're getting rid of our hydrogen this way. Okay? Yes? No? Now, why is it... Okay, if we're getting rid of hydrogen ions, what will happen to the pH value of that solution? It will increase, right? Higher the pH, lower the concentration of hydrogen ions. Okay? Hydronium ions. Now, this is one way. Our lungs help compensate. Okay? It's one of the fastest ways we can compensate for changes in our pH of our blood. Okay? And as you breathe, what do you breathe out? CO2. Okay? So, if you guys ever remember, all reactions, they're in some type of equilibrium. All right? Okay? And so, if you take all this away, what's it going to do? So if you breathe really fast, like if you hyperventilate, like next weekend before the test, and you guys are hyperventilating, what's going to happen to the pH of your blood? Go up. Why is it going to go up? If you're hyperventilating, you're getting rid of CO2. You're forcing this way. You're losing this hydrogen ion. A whole bunch of it. Okay? All right? So the pH is going to go up in your blood. Okay? You're going to be alkalotic, it's called. Not acidotic. Okay? We'll talk more about that later. But anyway, this is one main thing. And so this is, a big buffer system. There are also buffer systems inside our cells. On the other one, there's phosphate buffers that help regulate the pH in our cells. Okay? Also, proteins can act as buffers because they have groups that can act as acids and bases, suck up a hydrogen or donate a hydrogen. Okay? You guys good with that? We'll talk more about it, I promise. Now, last thing. Osmolarity. Now, you're probably like, why do I have to learn this? Okay? Well, let me ask you guys. So, any of you guys ever had some type of contusion or something else where you end up having edema, swelling? Well, what causes that? What causes swelling? What's that? Water retention? Well, it's retaining it, but where is it? What's that? Going into your cells? Well, where do we find most of our fluid? Outside. So it's outside our cells. So where's most of our fluid? Okay? Think about it. You start drinking water. It's going through your what? Circulatory system. Your kidneys are going to filter it out, right? Okay? Yes?
Right.
Okay. All right. And so, say you're drinking tons and tons of water. Okay? What's that going to do to your body? What's your body going to do? It's going to try to get rid of it, but also, what? You have too much. You have more water outside. Okay, your cells. So, let's see. I can draw this out. So it's just like this. So if inside the cell, it's very high concentration, and outside the cell, in the tissue, it's really low, where's the water going to go? It's all going to go into the tissues and cause that swelling. Okay? That's all it is osmolarity here, osmosis. All right? That solvent is going to go from one compartment to another, where the solute concentration is low, from low to high, to equal it out, because everything wants to be equal. Okay? All right? And this happens and so when you think about your blood, okay? All right? In your blood, what keeps the right pressure so that you're not too swelled with water or you haven't lost it all? You guys know? You guys ever hear of albumin? Yeah? What's albumin?
Blood component.
So what's that?
A component inside the blood.
So it's made by your liver. It's the main protein in your blood. And that solute, that amount in there, keeps the right, they call it oncotic pressure in your veins, okay? So that not too much fluid is coming in and not too much is going out, okay? So it regulates that where the water goes, okay? Cool, awesome, all right. Any questions about that? Should be good. All right, what else I got here? Now, a patient starts hyperventilating. Think about that. Okay, buffering system from the bicarbonate to the carbonic acid to CO2. What's going to happen to the pH of your blood? It's going to go up. Yes. Okay, remember that. All right. And then, how about if you're breathing too slowly? If you're breathing too slowly, it's the opposite, right? You're not getting rid of CO2. Okay? So that CO2 is going to force it the other way. Okay? All right, good. Cool. Molecules that have both hydrophobic and hydrophilic character are called amphipathic. Good. Awesome. Now, what functional groups are in the colored boxes? What's this group? The yellow. Aryl group. Good. What's this one? Primary alcohol. Good. What's this one? Secondary alcohol. Good. All right. Let's see. What are the functional groups in the purple ovals? Well, not really. I should have put not functional groups, but what are the bonds here? What are the linkages? What are those? Go ahead. Ethers, good. An ester would be what? Right? Okay, good. Awesome. Which of the following functional groups does ibuprofen contain? This is the kind of questions they'll have, this, just like this, on that nomenclature exam pharmaceutical foundations. What we got? Let's see. Do I hear an A? Okay, I got it. Got an A. All right, there's A. What else we got? Somebody. C, methyl groups, right? A whole bunch of methyl groups. What else have we got up here? We got an E. We got any E's? Any amines? Ethers? No. Carboxylic acid? Yes. Okay, good. Excellent. You guys would pass that test today if you took it. Okay. All right. Well, you guys got any questions? Remember, go back, look over it, especially if it, if it's been a little while, because I understand. All right, and just look it over. If you got any questions, write them down. Come see me. Okay. Remember, tomorrow class is at 10 o'clock. Okay. I don't know if you have class. You have class before it though, don't you? Okay. So yeah, we're 10 to 12 Tuesday, Thursday, and Friday over in Hartness. It's much smaller, and I can pick on you guys more. You have to sit close. All right, I'll see.